In 2008, the leaders of the CMTA were not satisfied with the current research operations. They came together as a single body to form STAR, the Strategy to Accelerate Research, with hopes of creating a pathway to the first treatments for CMT. Currently, STAR is pursuing pathways for 90% of all CMT. 1A, 2A, 2E, 1X, and 1B, and has identified a team of strategists for Type 4. Here I will show you what the CMTA is doing specifically for each type of CMT and define those first treatments. CMT1A is caused by too much peripheral myelin protein, or PMP22, which causes damage to the cells that make the myelin, or the protective covering of the nerves. Understanding the cause of CMT1A allows us to find a pathway to fix it. First, we had to make a line of cells with CMT1A, which we could use to test existing drugs. We created this cell line, which is now at the National Institutes of Health, testing 400,000 current drugs with several positive results. The cell line is also at Genzyme, a division of Sanofi, which is currently testing over 1.7 million of their compounds. While these cell lines were being tested, scientists were building laboratory models with which we could further test our candidate drugs. The models are robust and are currently testing several interesting compounds. And because we want to treat people with CMT, we've created a human cell line using adult skin cells to further test our candidate compounds. The CMTA's STAR initiative has partnered with investigators at University of Wisconsin, Johns Hopkins, University of Würzburg, and the New York Stem Cell Foundation to build robust human myelin cell lines with which we can test candidate drugs. The CMTA has partnered with numerous pharmaceutical companies to help us in our search for treatments. Our alliance with Genzyme will screen 1.7 million compounds against CMT1A cells. Addex has a compound which has demonstrated success in early stage testing. Our partnership with Addex will conduct further investigations. Through our work at the NIH's National Center for Advancing Translational Sciences, over 400,000 compounds are being screened against CMT1A cells. In a collaboration with ISIS Pharmaceuticals, the CMTA is working to develop molecules to improve CMT symptoms. And with Pfizer, we are conducting further exploration of 3,000 compounds. Through these multiple collaborations with our pharmaceutical partners, we are exploring numerous strategies to find viable treatments. And look at all the researchers committed to people with 1A. We have gathered a team of strategic, proven researchers who will help us find our treatments. But all of that work is just for CMT1A. Let's look at how STAR is addressing CMT type 2A. CMT type 2A is different from 1A in that the axon of the nerve, the primary nerve messenger, is affected. Fortunately, we understand this cause as a disruption in the proper ratios of MFN1 and MFN2, which prohibit the nerve from being a strong messenger. We have put together a brilliant team of researchers to build the cell line which we can use to find possible candidate drugs. Meanwhile, Dr. Bob Balow is building a human cell line from adult skin cells to create an assay with which we can test multiple candidate compounds very quickly. These tests will validate our findings. And like CMT1A, we need laboratory models with which we will use for further testing before we go to human trials. In 2014, Sage, Dr. Sperrin, and Dr. Stephen Shearer and the STAR team successfully built and is validating the first CMT2A laboratory model. This is a major accomplishment for STAR and for people with 2A. Clearly, the CMTA's STAR Scientific Advisory Board has been busy, but we still have more progress to share. STAR has identified CMT type 2E as a type worthy of early exploration because we understand how the nerves are affected. Like all type 2s, CMT 2E is caused by damage to the axon of the nerve's primary messenger. However, in CMT 2E, we are exploring the role of aggregates that form in the nerve cells. We believe that if we can find compounds which will either destroy these aggregates or prevent them from forming, we will prevent the damage to the axon. Doctors Liam and Messing are building a cell line to get the search for treatments underway. Meanwhile, the CMTA has contracted with Psychogenics to characterize Dr. Liam's CMT2E laboratory model for eventual testing of candidate compounds. This deeper understanding of CMT2E is a direct result of the STAR initiative. Another variant of CMT is 1X, a demyelinating form of CMT which is carried on the X chromosome, meaning that the gender of the person with CMT determines the severity and the inheritance patterns. Because males only have one X chromosome, they tend to be more severely affected than females who have two X chromosomes. Dr. Martini is working with CMT1X stem cells to evaluate the role of CSF1, which is believed to cause neural damage in CMT1X. In parallel, the CMTA, led by Dr. Shearer and Scheidler, is funding Dr. Martini's collaborative work with Dr. Shearer and a biopharma company which is testing compounds to evaluate the role of inflammation within the cell. We are very excited about the progress we are making with CMT1X. As you can see, the projects which were underway in previous years have accelerated immensely due to the CMTA STAR initiative. In 2014, we launched one new strategy for CMT1B. CMT1B is caused by a disruption in the unfolded protein response, which ultimately leads to the demyelination of the peripheral nerves. Drs. Shai, Wright, and Sverin have joined together to create cells which will lead to eventual assays to detect the desired outcome of CMT1B. Dr. Rabbits is creating a laboratory model with an identified mutation known to cause CMT1B, and an assay derived from human cell line developed from adults living with CMT1B is also being produced. And still to come is a pathway to attack the different types of CMT4. Strategies are being discussed with a team of the best CMT type 4 minds to look for a pathway for drug development. But we won't have a treatment ready for CMT until we have successes with clinical trials. The Rare Disease Clinical Research Network has joined forces with the Inherited Neuropathies Consortium to create a database of people with CMT. This database will be used to find those interested in participating in Phase 2 clinical trials. If you're interested in being a part of our database, please visit the website below or the Rare Disease Clinical Research Network's homepage to learn more and to enroll. We are fighting for you to find ways to treat your type of CMT. There are over 300 researchers and scientists dedicating their careers to you, but we can't do it alone. We need your help. Join our fight by giving generously to the CMTA. We will achieve our goal of delivering the first treatments for CMT with your help. Visit cmtausa.org/donate now to join our fight against CMT. Thank you.